Thank you. So we built this conference with the idea that we would start from the smallest scale moving outward. So we're going to be starting with the structural level of VCP. This session is led by Dr. Peter Shen. He's an associate professor of biochemistry at the University of Utah, where his lab uses structural biology and biochemistry to determine high-resolution structures of VCP and other protein homeostasis machinery. His group has produced key structural insights into how VCP unfolds its substrates through a hand-over-hand mechanism and engages with its cofactors, which has helped us to understand and define the fundamental principles of how this AAA plus ATPase fundamentally functions. Please welcome Peter Shen.
Thank you, Zali. We are a little bit behind schedule, so I'll be brief here in introducing our session. During the welcome session, we heard about the central challenge facing the field, in that VCP mutations cause a range of phenotypes, cause several diseases, including MSP, ALS, FTD, Paget's disease, but we still don't fully know why. Today's session is really about tackling that question at its most fundamental level. By understanding how VCP works at the molecular level, including how it recognizes substrates, how it interacts with its many binding partners and cofactors, adapters, and how disease mutations alter function, we can begin connecting genotype to mechanism. Those mechanistic insights provide the foundation for everything that follows, including identifying therapeutic targets and future translational research. So this meeting brings together patients and their families, clinicians, and scientists across the translational spectrum. And this session represents one end of that continuum. And so our first speaker is Dr. Andy Martin. He's a professor of molecular and cell biology at UC Berkeley and an HHMI investigator. And his laboratory studies the molecular mechanisms of protein homeostasis, including his seminal contributions in understanding how the proteasome works, along with many VCP complexes. And so today he'll discuss how FAF1 and other UBX-containing cofactors modulate VCP-mediated protein unfolding. And so please join me in welcoming Andy Martin.
So thank you very much. Thank you, Zoli, Peter, and Cure VCP Disease for the kind invitation. Really excited to be here at this inspiring meeting. And as Peter said, we are interested in basic mechanisms. So I will tell you about how VCP works, especially in the context of the proteasome or the ubiquitin proteasome system, because we heard from Chris that it has a role a lot in autophagy. And depending on its cofactors, it also works together with the proteasome. And to quickly introduce this, so I think many of you are familiar with the fact that the proteasome recognizes its substrates through attached ubiquitin chains. But it also requires unstructured flexible initiation regions that can go into the ATPase mode of the proteasome. And I think the pointing is a little bit of a challenge. Maybe I can point it out with my hand if you can see from over there. So the proteasome depends on these flexible regions in its substrates to allow degradation and mechanical unfolding and then cleavage into small peptides. But if proteins are well folded, and a large fraction of the cellular proteins are well folded, they may have ubiquitin modifications, but the proteasome cannot process those. So this is where VCP P97, or in yeast called CDC48, comes in together with its cofactor, one of the cofactor pairs, a heterodimer UFD1 NPL4. It recognizes these ubiquitinated proteins. It unfolds them, and then the partially or completely unfolded proteins are forwarded to the proteasome for degradation. And this is very important for general protein homeostasis, for ER-associated degradation, and other proteolytic processes. Now, how does it do this? How can VCP with this cofactor recognize these substrates that don't have any flexible handles hanging off of them? And really, seminal work by Tom Rappaport's lab a few years ago found out that the cofactor, and particularly the NPL4 portion that is shown here in this brown representation, picks one of the ubiquitins in the ubiquitin chain. It binds it in an unfolded state such that the N-terminus of this, what we call initiator ubiquitin, can hang into the ATPase motor of P97, or in this case the yeast homolog CDC48, and is then mechanically pulled on, and that initiates the unfolding of the ubiquitin chain as well as the associated substrate. Now, a former graduate student in the lab, Cameron, followed up on this to understand the mechanism of this in more detail. So he used a fluorescent, intrinsic fluorescent protein, EOS, to study the unfolding kinetically. So he put a ubiquitin chain on that EOS and then could follow unfolding by the yeast, CDC48, through the loss of fluorescence. And so he did this in what we call single turnover experiments and stop flow, rapid mixing to really get good kinetics, and had the EOS labeled with different length of ubiquitin chains, and found that when you have 5 or 6 ubiquitins, the substrate can be processed in less than 5 seconds. So there's very efficient recognition and unfolding of these substrates by the yeast homolog CDC48. So now, to get a better idea where the initiation happens, I told you that the system, the cofactor, grabs one of these ubiquitins in the ubiquitin chain. To understand better where initiation happens, he used a FRET-based assay. So he placed the fluorescent donor, Cy3, on the N-terminus of one of these ubiquitins, in this case the second ubiquitin in an unanchored ubiquitin chain. And he put an acceptor fluorophore at the bottom of the ATPase motor to then follow the initiation. So when the initiator ubiquitin is unfolded and sticks an N-terminus into the motor, we have the 2 dyes in close proximity and they transfer energy. We can read this out through FRET efficiency. So shown here is rapid mixing again to look at how quickly does the initiator insert itself into the ATPase motor. And we see it happens within about 3 seconds, so very rapidly. Interestingly, when he put the fluorescent dye on the first ubiquitin in the ubiquitin chain, he didn't see this FRET signal develop, meaning that the initiator has to be at position 2 in the ubiquitin chain or higher up, and you need additional ubiquitins, at least one, on what we call the proximal side to allow this process to happen. So the question was then, how is this, and what we call a lysine 48 linked ubiquitin on the proximal side, highlighted here in red, how is this sensed by the system? And we had proposed that a domain called the UT3 domain of the UFD1 cofactor, part of the UFD1 NPL4 pair on top of CDC48, is responsible for recognizing this red first ubiquitin here. And because we didn't have structure information, you used AlphaFold for this that predicted how this proximal ubiquitin may bind to the UT3 domain. And it was a very reasonable model that what we call the hydrophobic patch around I44 and ubiquitin makes contacts with the UT3 domain around this leucine 55. And we could then also confirm this by removing this leucine 55, mutating it to an arginine, and disrupt this interaction of the proximal, this red ubiquitin there, and see that in our FRET-based assay, we no longer see the initiator efficiently insert itself. So you need the red ubiquitin bind to the UT3 domain in order to the neighboring ubiquitin being able to insert for initiation. So to summarize this early work, the UT3 domain binds the proximal ubiquitin in a lysine 48 linkage-specific manner in the ubiquitin chain and facilitates unfolding and insertion of the neighboring initiator ubiquitin very rapidly within 3 seconds. Now, what about P97 VCP, the human homolog of this? This is what a current postdoc in the lab, Sang Wai, took on. And we were aware that P97 with its UN cofactor is known to be very slow in substrate processing. So to be able to do quantitative studies, he slightly modified the substrates, and some of you may be familiar with this. This green fluorescent EOS can be photoconverted into a red EOS version that has a split backbone. And the advantage is then if motors like P97 or VCP work on this substrate, they separate the 2 parts of this EOS, now split EOS substrate, and they irreversibly get separated and they can no longer reform later. So you get irreversible unfolding and irreversible signal change of red fluorescence. And that's what he monitored here, and again in single turnover kinetics, and found that it takes a P97 with UN cofactor almost 80 minutes to unfold the substrate. So it's very slow. It's about 3 orders of magnitude slower than the yeast homolog CDC48 with the UN cofactor. So we thought, what is rate limiting? Maybe the initiation, picking the right ubiquitin and inserting it into the motor is a rate limiting step. And so we knew of previous work by the Labib lab that described a cofactor FAF1 that reduced the ubiquitin chain length threshold of P97 UN. So meaning in the context that they studied replosome disassembly by P97, they found that where you have shorter ubiquitin chains, FAF1 can help initiate. So we thought maybe it helps initiation. And this is what FAF1 looks like. It has an N-terminal UBA domain, this small thing here. It has two ubiquitin-like domains in pink and in light blue. It has a US domain that may be involved in polymerization of this. And then it has a long helix, here shown in cyan, and a C-terminal UBX domain. So when Seng-Wai added this FAF1 to our unfolding reaction, he saw a moderate but still 5-fold acceleration of the substrate unfolding. And he then did deletion studies to see what is necessary and sufficient in FAF1 to allow this acceleration of P97UN-mediated unfolding. So he cut off different domains along the N-terminus and found that basically just having this long helix and the UBX domain is necessary and more or less sufficient for having this accelerating effect. He also then tested a homolog of FAF1 that's called FAF2. This is a membrane-bound homolog. It doesn't have the ubiquitin-like domains, UBL domains, but it has a membrane anchor, so it works in other contexts. But similarly, we see acceleration. And again, the long helix and the C-terminal UBX domain are sufficient to have this acceleration in a VCP-mediated protein unfolding. So, but how fast is this initiation actually happening? Is it really making it much faster to pick the ubiquitin and stick it into the P97 motor? For this, he used this FRET-based assay that I already introduced to you. So he used unanchored ubiquitin chains again. He labeled the second ubiquitin with the Cy3 donor fluorophore, attached acceptor fluorophore LD655 at the bottom of the P97 motor, and then analyzed the signal changes. So first he looked at spectra. So shown in red is the spectrum of the donor-labeled ubiquitin chain. In blue is the motor with the acceptor label. When he mixed the two, in the presence of UN, you see it's basically the sum of the two spectra. There's nothing happening because the initiation is so inefficient with just the UN cofactor. It does not allow efficient insertion of the initiator. But when we added FAF1, now you see a typical FRET behavior where the acceptor, the donor fluorescence goes down, the acceptor fluorescence goes reciprocally up. So there's a FRET exchange, meaning that there's efficient insertion. So we have a scenario like this. So he then also tested the fragment again. So instead of full-length FAF1, he used just this helical fragment and the UBX domain, and that also gave a similar signal change. So the fragment is sufficient for activity. He then tested this in the presence of the non-hydrolyzable ATP analog ATP gamma S to prevent translocation. So you would just see the initial insertion, and then the motor would not be able to pull any further and pull these proximal ubiquitins through the central channel. And interestingly, he didn't see a robust FRET signal change there. So meaning the FRET development actually depends on ATP hydrolysis and the translocation activity of the motor. And this can be explained in this scenario. We have the fluorescent dye only attached to the second ubiquitin. So to get a signal here that is independent of translocation, you would get this only if really the second ubiquitin that carries the dye is chosen as the initiator. If an initiator higher up in the chain, number 3, 4, or 5, is picked as the initiator ubiquitin, then you wouldn't see a signal because shown here, for instance, the red ubiquitin is number 4. If that is chosen as the initiator, it wouldn't give a signal until ATP-dependent translocation pulls in this proximal part that actually carries the fluorescent dye and brings these dyes into proximity to get a FRET signal. So therefore, we think that higher ubiquitins in the case of P97, UFD1, NPL4 are chosen as initiators, possibly because the UT3 domain of UFD1 is binding maybe 2 or even 3 ubiquitins. So that was a first interesting indication. He measured kinetics, so going back, how quickly does this happen? With the help of FAF1, now P97UN can initiate in about 5 seconds. So it's much faster. It is 3 orders of magnitude accelerated when FAF1 is present for unfolding. So for comparison, again, P97 with UN is very slow. It takes about 5,000 seconds, and initiation is rate limiting. If we have the FAF1 cofactor helping, we now are in the 5-second range. It's very similar to where the yeast homolog CDC48 is with its initiation, even though then unfolding itself is still slow for the human homologue P97 compared to the yeast homologue. What does this look like? How does FAF1 does its job? How can it accelerate initiation so well? We did Cryo-EM structure determination. We had the P97 motor in the presence of ATP, the UFD1 NPL4 cofactor, and with this FAF1 fragment of the helix and the UBX, and free ubiquitin chains. So we saw basically the UN cofactor here in green and blue, and the ubiquitin draped over it. We see a couple of proximal ubiquitins, the initiator ubiquitin bound to NPL4, and these distal ubiquitins that are higher up in the ubiquitin chain. Now, to zoom in here, first, looking at the UFD1 cofactor, it has an N-terminal UT3 domain, and then it has two what we call ship boxes that are separated by the NPL4 binding motif. And if we look at the structure here, we see the two ship motifs, two ship boxes bound to two neighboring N-terminal domains of the P97 hexamer. We see the NPL4 binding motif bound as expected to the NPL4 cofactor, and we see the UT3 domain hovering up here. It doesn't make any direct contacts to the P97 motor or to the NPL4 cofactor. If we now look at the NPL4 cofactor part, it has an N-terminal UBX domain and then two zinc finger motifs, a ubiquitin binding MPN domain and a C-terminal domain. So we see the first zinc finger, CF1, bound to the ATPase domain of one of the protomers of P97 hexamer that we call protomer F. If we turn it around, we see the UBX domain bound to the neighboring protomer of P97, to the N-terminal domain. And then we see the second zinc finger, CF2, bound to another ATPase domain of protomer D. So in this schematic, you see the occupancy of these cofactors on the P97 hexamer. So NPL4 occupies two ATPase domain and protomer F and D, and the UBX domain binds the N-terminal domain of the P97 protomer E. And we have the two ship boxes of UFD1 binding to two neighboring N-domains of the protomer B and C. And the same N-terminal domains are also occupied by two copies of FAF1. So we see two FAF1 cofactors bound, and they bind to the same N-terminal domains that are predetermined by the UFD1 cofactor through binding of the ship box. So now going to the ubiquitin, what do we find about ubiquitin interactions of the whole system? We see these distal ubiquitins, three of them bound to the tower binding sites of NPL4, so that is on top of the NPL4 cofactor. We see the initiator ubiquitin, shown here in salmon, bound to the hydrophobic groove of NPL4 in an unfolded state, and it reaches with its N-terminus into the P97 motor. The first residue of this initiator ubiquitin is lysine 11, but there are 10 extra residues that hang into the bottom part of the P97 motor, into the D2 ring. We have high enough resolution to look at the nucleotide occupancy of the D1 ATPase ring and the D2 ring. We see 3 ATPs and 3 ADPs bound in the top ring, and we see 3 ADPs bound in the bottom ring. So it means that the D2 ring is in an all ADP bound state. We think that this is still in an initiation state. It's not yet the spiral staircases that Peter nicely described with his structural studies. So we think that this is before engagement of the pole loops, that they start pulling on the initiator and go in hand-over-hand translocation. This is still in a kind of an initiation pre-engagement state. So what was exciting was that for the first time we saw the initiator ubiquitin bound to NPL4 also having this bridging density over to the UT3 domain. So you see this over here, this linking density. And if you turn it around, the last 7 C-terminal residues of the initiator ubiquitin are bound to this groove of the UT3 cofactor. And so the C-terminus ends right next to the lysine 48 of another, like a proximal ubiquitin, meaning that there's a covalent, there's an isopeptide bond there, as you would expect for K48 linked chain. And this UT3 domain bound to these ubiquitins is stabilized by the FAF1 helix. So FAF1 is bracing this ubiquitin-bound UT3 domain and anchoring it to P97 through the UBX domain binding to the N-terminal domain. So if we turn it around, I mentioned there are two copies of FAF1, with FAF1A and FAF1B, that bind to neighboring N-terminal domains, and they actually seem to brace the UT3 domain from two sides, almost like a tripod here, and stabilize the UT3 domain during this initiation process. And even though we don't have a very strong density for the second copy of FAF1, we do see different classes in Cryo-EM that all have both of these legs for two FAF1 copies. We see the UT3 domain in slightly different positions, wobbling around, but overall, it seems to be stabilized by two of those legs. Now, the interaction of the second FAF1, FAF1B here, is either directly to the UT3 domain, so it may make direct contact with the helix to the UT3 domain, or it may be mediated through an additional ubiquitin. And we think this because there's extra density here, and in the middle of the density, the proximal ubiquitin that is linked to the initiator has its C-terminus ending right there. So there could be a second proximal ubiquitin localized here that then is braced by the helix of the second FAF1 molecule. So we get much better idea of what the first FAF1 copy does here. It binds directly to the UT3 domain. So if we flip it around, it braces that UT3 domain. And AlphaFold help with more detailed view. We have especially three residues, a tyrosine, serine, and then a aspartate, docking into the surface features of the UT3 domain. And we tested this interaction by disrupting it. So for instance, we mutated the serine to a lysine, or we mutated the alanine UT3 domain to a larger residue asparagine. We can disrupt this interaction, and we no longer see an acceleration of unfolding. So how does FAF1 now help initiation? So in the presence of FAF1, we see that this C-terminus of the initiator ubiquitin binds much more deeply into UT3 groove. And AlphaFold, again, helps when we use AlphaFold to look at binding of this ubiquitin to the UT3 domain. Initially, you see that the ubiquitin has its C-terminal beta strand extracted, and the ubiquitin is strongly destabilized. So this is in comparison to the scenario in the absence of FAF1, where we see ubiquitin normally bound. So FAF1 helps destabilize the initiator ubiquitin. And you see this also here. The initiator ubiquitin is strongly destabilized. We think that this is the initial scenario when ubiquitin lands on the system, on these cofactors. Ubiquitin binds in that groove. With the help of FAF1, it gets destabilized. And then it can bind NPL4 and find its way into the motor, into the central channel of the P97 motor. So to summarize this part, I showed you that when the ubiquitinated substrate lands, FAF1 stabilizes the UT3 domain. It helps destabilize the initiator ubiquitin by forcing it into this deeper groove. This destabilized ubiquitin then will unfold and bind to the NPL4 cofactor and stick its N-terminus into the motor, and then this allows initiation of substrate processing and unfolding. I want to highlight that similar structural and biochemical results were also reported by other groups. And I'm running out of time, but at the end, I just wanted to summarize again. We have initiation within 5 seconds. Unfolding still takes 15 minutes. So this is 200-fold slower than the yeast system does it. And so a possible reason is that the P97 VCP, the human enzyme, works much more slowly in ATP hydrolysis than the yeast enzyme. It's 60 times slower. But are there other cofactors that could further accelerate this processing? One candidate is P47. It has been worked on by Hemmo Meyer for quite some time. Peter had done some structural work on this, but we still don't know how exactly it works. It also has a C-terminal UBX-domain. So it may occupy some of the still open N-terminal domains. We have 3 N-terminal domains that are not occupied by the UN and FAF1 cofactors. So there may be a simultaneous or consecutive action of that cofactor together with UN and FAF1. And we tested this. We added P47 to either by itself to the UN P97 or in the combination with FAF1. And we see it accelerates it, but we don't really know which stage it does because it has accelerating effect on all these different processes. So P47 may affect initiation, initiator engagement, unfolding of the ubiquitin chain, or unfolding of the substrate itself, and I can't give you the answer yet. But we see a potentiating effect together with FAF we see now a 10-fold acceleration compared to having just P97 and the UN cofactor. It is still much slower than the yeast homolog, 50 to 100-fold, and maybe this is where the 60-fold lower ATPase activity is to blame, that the human motor simply works more slowly than the yeast homolog. And I know I'm out of time. Just real quick, linking it back to MSP mutations, I think you're familiar that many of them are at the interface between N-terminal domains and the ATPase domain. And we wanted to see, do they differentially affect these cofactors? Do we see stronger phenotypes in our in vitro studies? So one of the mutations we tested is this alanine 232 glutamate that has been previously shown to affect N-terminal domain positions and moving up and down in response to ATP hydrolysis. And also it accelerates the unfolding of substrate by P97UN. But when we tested this in the presence of FAF1 or P47 plus FAF1, we see consistently this twofold acceleration. So there's no differential effect on these different cofactors. And therefore, we think that disease phenotypes may originate from differential cofactor binding that are not UBX-domain containing proteins. So all of these cofactors are similarly affected, maybe because they all bind similarly to the N-terminal domains, but there are other cofactors that bind differently, and they may then be differentially affected by these MSP mutations. So this is just the summary of what I told you, but I want to highlight the people who have done the work. Seng Wai was responsible for most of the structure function studies with the help of Nitin, who did the P47 studies. So thank you very much.
Questions?
If you do have questions, please wait for a mic so we can get it on the recording. Here you go, Virginia.
Thank you. Virginia Kimonis. That was very elegant work. My question pertains to how does the hypothesis that gain of function is the cause of the disease fit in with your hypothesis?
Yeah.
Or with your work?
I was
yeah.
alluding to this. I think it's still unclear whether it's gain or loss of function, because if these mutations affect distribution of cofactor occupancies, like especially because many of them are in the N-terminal domains or in the linker or the interface between the N-terminal domain of P97 and the ATPase domain, then these differential movements may affect which cofactors bind with higher affinities. And Chris mentioned that certain PLA8s, certain other cofactors are not binding anymore. So you may have a shift in these cofactor occupancies of VCP, and therefore a shift in how much activity you have in these various different roles that VCP has in the cell. So you may, by increasing activity in one aspect, you may lose out activity in another important aspect. And because it's such a complex mechanism, or the tasks are so various, and you have a complex disease phenotypes, it's still unclear what is really the reason for this. Is it do we need an inhibitor of P97 or VCP, or do we need an activator, or do we need something that controls N-terminal domain positions depending on the mutation that is present in these different diseased variants? So we don't know. We were hoping to see differential effects on FAF or P47 with this mutation, that maybe some effects go up, others go down, but we don't have good insight.
Andy, great talk. Just a question about past initiation to go to actually substrate processing. Do you think FAF has to let go? The cartoon you drew dissociates. So do you think, right, as the substrates go through, would you have to unbind?
So we tried to address this with single molecule studies. It's still unclear. We don't know which cofactors, what the residence time is, how they come and go. We have, it's unclear. We have some preliminary data, but it's tricky. Yeah, it's. There are so many other cofactors and deubiquitinating enzymes that may come and may require some of these delivering cofactors to leave, but we don't have a good idea about the temporal kind of distribution and what comes and goes at what time. Yeah.
Is there time for a question?
Sorry.
So then for FAF1 and FAF2 then, do you think that the threshold is like greater than 5 ubiquitins. So what cohort of cellular substrates actually need these two cofactors for degradation?
I mean, that's a good question, because when people look at length of ubiquitin chains, K48 linked ubiquitin chains, they are usually they're not too long, they're not beyond 10 on average. So it may be then through the help of these cofactors. It was just in vitro, you think that you need these extensively long chains of 15, 20 ubiquitins, but with these cofactors, you bring it down. We see very robust unfolding with things that are at 10 or below 10 works just fine. So yeah, but it's still a little bit unclear how this is managed and how ubiquitin ligases extend this and dubs all shrinking it again. And so it's linking the in vivo scenario with what we do in vitro can be challenging in that respect. Yeah. Okay. Sorry, my fault. It was too long.
Sorry, yeah. I just wondered if you could say a few words about the druggability of the complex as you understand it now. Do you get any new insights from these cofactors? Do they give you new opportunities?
I mean, I think this would be something for later presentations, because we don't really look at the interfaces or to what extent you can. I mean, certainly you could find compounds that interfere to some extent, but what makes it harder is that many of these cofactors bind with their UBX domains to these N-terminal domains, and they all share similar interfaces. So if you found something that disrupts this interaction, it would affect a lot of different cofactors. So I think you would have to fine-tune, but I think Tarun Kapoor will have maybe answers to more sophisticated ways of addressing this. Yeah.
Thank you, Andy. I should also add that there will be a few minutes at the end, towards the end of the session, where all the speakers will be up here for a short panel discussion. So we'll have additional Q&A at that time. Our next speaker is Dr. Po-Lin Chiu from Arizona State University, where he's an associate professor there, and his lab uses Cryo-EM and Cryo-ET to study structures of very dynamic complexes, including VCP. And so today he'll tell us more about the allosteric regulation of VCP. So please join me in welcoming Dr. Chiu.
All right, good morning, everyone. And then it's a real pleasure here. And then I would like to thank Peter and the organizing committee for kind invitations and for giving me an opportunity to share my work on the VCP. So my lab is interested in understanding about the allosteric regulations of VCP, and it will primarily use Cryo-EM to study structure and mechanisms. So here just briefly shows that depending on the samples in different conditions, in crystals or in solutions or in the cellular environments, and we can use different Cryo-EM methodologies to determine the structures that we are interested in. And then the structure I'm going to show today is all determined by the single particle reconstructions. So basically the method is that we image the multiple projections of the molecules and then computationally reconstruct the 3D charge density map from these images. And so using these methods, the advantage is that we can set up multiple conditions by engineering different P97 mutants or use various ligands or cofactors, and then to assemble it into the solutions and then visualize the resulting structures using the Cryo-EM. All right, here is just a very quick review of how the VCP look like. So you have the Walker A and Walker B responsible for binding and catalysis, and then sensor 1, and then also at the polymer interface, you have arginine finger and intersubunit sensor units, and then to control the ATPase activities. And then go up, you have the D1, D2 linkers, and then you have the D1 ATPase that gate the D2 ATPase activities. Then it links the upper, we have the ND1 linkers, and then that links the NDD domains. All right, so we know that the D2 is mostly responsible for the VCP ATPase activities, but the P97 activities is influenced by the entire protein conformational change. And then because all these domains are mechanistically coupled and then through the flexible linker. And then so one example is that you see, you can see the NTD. The NTD can undergo transition up and down, and then also associated with the D1 nucleotide states. And you have the cofactors that can interact in the N-terminal domains or interact with the C-terminal domain to impact the ATPase functions. And also, we know that the disease mutations happens in the VCP that can cause the alterations of the ATPase activities. And then mostly the mutations is clustered in the NTD or ND1 linkers. So that tells us that if you have a structural perturbations very far away from the D2 domains, and then somehow you have some kind of pathway that you can propagate through the protein from the very far away NTD and then pass through to the D2 ATPase. So this raises us two questions. One, what is the key allosteric nodes that we know through this signaling pathway? And then second is that could we map that and then within these P97 molecules? All right, so the first testing condition is that we because we start from the NTD, and then so we take the R155H mutants, and then with the P47 cofactors, and assemble it in the solutions, and then use the Cryo-EM to study the structures. So here shows a typical Cryo-EM image, and then we can pick the particles from the image, and then classify them into different groups. And then based on this classified image, we can calculate the 3D Euler ingles to make the charge map densities. So we have two different kind of assembly show up in the populations. One is the dodecamer, and then that is the two hexamer of the P97 pack opposite against between each other. And then you have another structures that you show an hexamer, and then with a P47 bonds onto it. And then for the dodecamer, that we don't have any densities for P47 or for the nucleotides. So we presume that it's an apo state. And then it has NDD in the up positions. And to test that whether this dodecamer formation is caused by the nucleotides conditions, we use the Cryo-EM to do the assembly conditions in the presence of different nucleotides. So we can have the sample in the presence of the ADP, and then have the sample presence in ATP gamma S. Then we take a bunch of the images and to calculate the populations of these dodecamer and hexamer. And then we find out that if we don't apply for the nucleotides, then you see most of the dodecamer just form in the solutions. And then that's presumably like a storage form that does not form in the functions. But once you apply the ATP or ADP, then you'll see that the dodecamer populations dramatically reduce to almost none. And then we also do mutations to test this, is that we use these mutants and then to do mutations on the Walker A, and then to have the molecules enabled to bind the nucleotides. And we see most, around 90% of the populations are on dodecamer. And then if we look at the hexamer where the P47 binds onto this, on this VCP complex, we found that we don't have too much of the conformational change that we can detect from our experimental data. But we found that the D2 arginine fingers has somewhat have the conformational change. So that led us thinking about whether we can do mutations on the D2 arginine fingers to test how the function is altered. And we found out that the mutants, the arginine finger mutants with both on the wild type and also on the R155H, we found out the mutant is actually impact more than the wild type. And then if we test, by adding the P47, we found out that it's the P47 cofactor is much more impact on the D2 arginine fingers. So we kind of have some kind of chain that link from the P47 binding on the NTD and then go through to the D2 arginine finger. That's a very long process, long way from the NTD to the D2 arginine fingers. And then from the previous literature, we know that there's one allosteric nodes that is also a key to regulate all these pathways. It's from it's at the D1, D2 linkers. That's a leucine 464. So we also did the mutations on the L464 into different residues and test its functions. And then most of the residue does not respond, does not have the ATPase alter very much. Only the Po-Lin mutations cause the alterations of the ATPase activities. And then so if you look at the histogram, you'll find out that the L464P is reduced on both of the mutants that we test. One is without the D2 domains, and then one is the full length. That means that if you mutate the D1, D2 linkers in the middle, it actually affects bidirectional. It also affects the D1 ATPase as well as the D2 ATPase. So we take the protein assembly to on the Cryo-EM to do the imaging and then make the reconstructions. And we find out that the NTD configurations is actually heterogeneous. It's like a ladder. So you have the down states and then go up as an up state. And then if you also plot up the local resolution that we can use it to verify the flexibility of the D1, D2 linker, you'll find out that when the NTD is on the D down states, the D1, D2 linker is much more rigid. And then when it goes to the up conditions, you have the D1, D2 linker much more flexible. So you have some kind of like NTD couple with the flexibilities of the D1, D2 linkers and then affect the ATPase function. And then the other notes for the Po-Lin mutation is that Po-Lin is a very specific amino acid in the canonical amino acid set because it has a different up and psi and phi torsional angles. So it causes much more rigidities in the D1, D2 linker. So when we think about, when we mutate from the leucine to Po-Lin, we think about we introduce some kind of a geometric constraint on the D1, D2 linkers and make it much more rigid and incapable of doing its regular functions. So if you look into the details, we can see a little bit of shift on the D1, D2 linker in the structures. And then it caused that there's a helix-turn helix domains in the D2 domains actually move a little bit. So to answer these questions, why these small movements can cause the ATPase activities reduce, we can just search in some kind of like allosteric inhibitors and then try to minimize the movements of the VCP and see if we can have the function activities reduced. So what we test is the triazole-based inhibitors that we use. And so NMS A73 is one of the early report highly potent allosteric inhibitors. And then, but it has some limitations. One is that it has the poor solubilities. And also it has a poor PK properties and also has an off-target effect. So I collaborate with the teams in the CBC, NCI CBC next teams. And then Donna lead the project and then design the triazole class inhibitors. So we tested different types of inhibitors and then bind with the P97 and then to study its mechanism and function. And then so the design concept is that we maintain the tricyclic base and then we attach with the difluorobenzene rings and then also with the variant substituents in the triazole-based class inhibitors. So the binding site is sandwiched by the D2 and then adjacent D1s. And then the stabilizing force for the compound is by the hydrophobic interactions. And then we also identify two hydrogen bondings and for asparagine 616 and lysine 512 to interact with the tricyclic base. And then here shows that we have a slightly structure change when the triazole inhibitor binds onto the binding pockets. Like we have the one of C-terminal loop in the alpha 2G helices, and then it flips. So you have the two arginine flipped on the different sides. I'll show you the movie later. And also the helices the surrounding helices of this binding site actually also move. And then the purpose is to accommodate the volume expansions when your inhibitors occupy these positions. So to appreciate the movement of this loop change, so I morph the two structures. One is the bound states, one is unbound states. You'll find out that the loop is actually flipped from one end to the other end. So these local movements actually cause the residue side chain rearrangements in the protomer interface. And then if we compare the NMS A73 structure side by side, if we just first focus on the binding site, you'll find out that in the NMS-A73, it seems to have a very similar binding patterns for the surrounding residues. But you don't have the asparagine 616 and lysine 512 to engage with the hydrogen bonding. And then one of the likely explanation is that because the compounds that we use, the 462, compared to the A73, we have a longer compound's length. It's about like 6.1 angstroms. So when it is longer, then it's easy to well position in the binding sites. So allows the two hydrogen bonds to interact with the tricyclic base and then to stabilize the inhibitors. And if you compare to the polymer interface, that we have the two arginine fingers, and then with the ISS and subunits, it's more clustered and networked together with the Walker B motif. And this away from the nucleotides binding sites. So this might be one of the reasons that it inhibits the ATPase activities. But compared to the NMS-873, it shows a different conditions. You have the arginine finger 635 interact with the nucleotides instead of the Walker B. So here we don't know whether this is the reason of because the different nucleotide states, but now it seems that we can provide two different kind of nucleotide states and how their inhibiting modes look like. And we also test on the mutants, and then we use the different triazole-based inhibitors. And then the interesting thing is that we test 515 and 701 on the VCPs. And then usually from our previous experiments, we test with our 155H structures. The NDDs always shows in up configurations. But if we test with the 515, we all shows that the NDDs are up. But when we test with the 701, you have the up and down in the same populations. So using the Cryo-EM to visualize the structure change might be helpful to let us know what's the mechanism to control this NDD up and down. And then so if you superpositions of all the structure together, one of the critical things is that we test the compounds in the different fluorine substitutions. So if we instead using the meta substitution, we use the para substitutions. Actually, the fluorine in the para positions will have less electron withdrawing, so it maintains more electrons in the benzene rings. So in this way, it can maintain the stabilized interactions between the lysine residue, the positive charge, and with the ring. So it's the pi-cation interactions. And then the other properties of this 701 is that you have the 1, 2, 3 triazole rings. And this triazole ring provide a larger polar surface. So it can be more hydrophilic and then provide more potential hydrogen bonding acceptor sites. So here, if you compare the rearrangement of the residues, one notable structural difference is that the lysine 615 is actually changes conformations to interact with the triazole inhibitors. And then because the 701 have the different side chains on the compounds, it creates two different conformations in the binding sites. And in these ways that you have the different arrangements on the interactions of the compounds with the surrounding residues. And then because of this different arrangement with the hydrogen bonding network, you actually have the conformational change of the big D1 helix turn helix and to interact with the NDD. So you have the up conditions, you have the down conformation of the NDD. So what we learned from the previous experiment results that we know that since you have a small movement that you cause the VCP function change. So is there any way that we can use the existing all the structures and then use those informations together and then to learn in some kind of model that we can plot out the allosteric signaling pathway. And then one thing is that every experimental determined structures, it has some kind of informations, and then it gives you some kind of graph. And then so you can use the residue as a node with its property, and use the interactions between the residues as an age. So use the nodes and age to create some kind of graph and some kind of model for us to learn the allosteric regulations. So the method we use is the graph neural network, and then we take out all the structures from the PDBs and then try to learn. The ultimate goal is to learn latent representations for conformational landscape of the VCP, and then trying to learn from there and then to plot out all the allosteric signaling pathway. And then we are not including the structural informations. We also take out the functions from the papers and then to put it into the BESL as features in the training. And then this is the preliminary data that we show. All the structures lined up in the UMAP latent landscape is continuing rather than the discrete states. And then here just shows the preliminary data. So we have the model all shows that NDD has a rigid body movement. This is probably already the community already know. And then associated with the glutam-rich motif, and then the torsional and then structures are all associated together. And then we also do the perturbations response scanning and also the GNN explainer decoder, and then to find out 4 extra residues that can control the allosteric signaling. And then we also do the integrated gradient attributions to find out there's a highly negative charge motifs in the C-terminus that can control the dodecamer formations that also contribute part of the allosteric pathway. So once we have all this information, our next step is to use this residue to guide the molecular dynamics that we can learn how the signaling is propagates through this pathway, and then along with the time. And we also will plan to do the experimental validations on these identified residues. All right, so this is the last summary slides. So this is what we learned from the assembly with the P47 with the mutants, and also the D1, D2 linkers play a very essential roles in the allosteric regulations. And we also test with the triazole inhibitors. And then the next step is doing the MD and also the experimental validations on our allosteric signaling pathway. So for these studies, I would like to acknowledge my students, Prabhasha and Edgar. And then I also thank DY for the help on the Cryo-EM data collections. And I'm also grateful to have a long-standing collaboration with Tuifan and also to her lab members. And I also want to acknowledge Donna, Mark, and Andrew for our collaborations supported by the CBC next project. Thank you very much.
Hello. Thank you so much for your talk. That was great information on allosteric inhibitors. I actually had a question about the dodecamers that you see. You mentioned that when you add ATP, you see the change in population from mostly dodecamers to mostly hexamers. And I was wondering if you had any hypothesis of how that mechanism of dodecamer splitting occurs, and like how much time did it take after incubation with ATP to switch those populations?
I don't have the exact studies that we learn from the cells or in the presence of other proteins, but I have in the lab, we have other studies working on other AAA ATPase. And then it's in plant. So it has a very similar trend that when the AAA ATPase is not used, they kind of stagger each other. And then when it needs to be used, the cell change the ATP-ATP ratios, and it start to get disassembled. So this is basically what we can hypothesize that why the dodecamer could be detected in the vitro conditions. But maybe this is just an artifact that we see in the vitro that we don't know. It's just that this is our observation.
Thank you.
Yes.
Thank you for the talk. So I wonder, how does the heterohexamers respond to these conditions? Is that from the beginning compact, or does that respond to ATP appropriately? How does that work?
So in the very beginning of the experiment, we didn't supply any of the nucleotides. We just basically just assemble the two proteins. So you're asking the dodecamer, right? The ratio.
Correct. If you have mutated ones and normal ones together, how those assemble to begin with?
That we have not tested. We don't know. Yeah. That's a good idea. Thank you very much.
Okay, thank you, Dr. Chiu. All right, in the interest of time, we do need to move on. And so the final speaker of our session will be Dr. Sriram Subramaniam from the University of British Columbia, where he is a professor of biochemistry, as well as founder and CEO of Gandiva Therapeutics. Sriram probably doesn't need an introduction with this crowd. He's done a lot of foundational work in terms of VCP structures, including drug-bound complexes, and today he'll tell us more about using AI and structural biology to advance our development of VCP-targeted therapeutics. Sriram.
Thank you very much, Peter. It's really an emotional experience for me to listen to all of you, and it just inspires me to do more, to accelerate, to get to a cure. And I just want to begin with really the end game here, which is we want something that's approved by the FDA. And so I think beginning with the end in mind is always a good thing. And if you look at the numbers, FDA approves on average maybe 50 or so drugs a year. So if you look in the last 2 years, 100 new drugs were approved. So keeping that as a goal is always important, and obviously we're not there for numerous diseases, and certainly VCP P97 that's in that league. So now, if you look at what kinds of drugs are approved by FDA in this set of 50 or so annually, oncology is the major contributor. It's about a quarter plus are oncology drugs. There's various other diseases. But what's interesting here is that the majority of even the oncology approvals or the others, they target single proteins, meaning they're typically kinase inhibitors or things that go to the catalytic or the active site. But if you look at how biology works, it doesn't really work like that. And there's a second issue, which is that, this was touched on earlier this morning, which is it takes really long. It takes 10 to 15 years to get to the market, and this is from the time that a patent is filed. Someone can develop it. So these are the real challenges as we go forward. But I want to touch on the scientific challenge, which is the biology, be it VCP P97 or pretty much any other disease, fundamentally is driven by protein dynamics, proteins moving around, and transient protein interactions. So, and essentially, if you were to look inside the cell, if you were to walk into a cell, this is probably what's going on. You have proteins binding to each other, coming off. There are transient interactions. Some proteins are changing shape. Some proteins are actually getting shut off. They're coming back on. So our task really is, how do we deal with this complexity, and how do we actually address specifically the proteins that we care about? So I want to share with you a couple of examples today from this overall landscape of how we're thinking about going after drug discovery. There's many other sessions later in this symposium that touch on that. And so I just want to touch on mostly on the role of VCP P97 in a newly discovered sort of role in Adams-Oliver syndrome. This is work with colleagues at the University of British Columbia, medical geneticists who found this in a pediatric manifestation, and some ideas about how we might design inhibitors that compensate for some of the liabilities that are seen in the clinic. Time permitting, I'd like to very briefly touch on how we're thinking about designing small molecules that target protein-protein interfaces. These are called molecular glues. It's just what the word means, which is we're trying to find molecules that glue together interactions so they cannot do what they normally do or deal with this function this way. And finally, just a couple of slides on Cryo-EM technology, which you already heard a lot about today, and where it might be headed as we look to the future. These are slides you've seen earlier today, and about 10 years ago, we began to work on the structural biology of VCP-97. We published the structure of the hexamer, and importantly found out that the binding of ATP leads to this dramatic conformational change in the green portion, which is the N-terminal domain. So the binding of ATP, which is to the lower, the D2 domain at the bottom, leads to this big change in the N-terminal domain. And in subsequent work, we found that many of the mutations in multisystem proteinopathy uncouple this conformational change. In other words, the change normally requires the binding of ATP and the hydrolysis of ATP, actually, or just the binding of ATP, I should say. But in these multisystem proteinopathy mutations, this change is uncoupled. It does its own thing. More recently, and this is work that we've done at the University of British Columbia, my colleagues Anna Lehman and Milan Patel in the Department of Genetics have been seeing patients that have this pediatric disease called Adams-Oliver syndrome. And a subset of them have pulmonary hypertension. And from careful analysis, they found that these patients had mutations in VCP-97, which was a surprise because normally this is not the kind of patient cohort that they expected to see these diseases. And typically the manifestation is in terms of limitations in limb growth and several other anatomical features. And this is a pediatric disease now, which is very surprising for VCP-97. And what is even more striking is that if you look at the location of these mutations, they're all in pretty much the same kinds of places we've seen in other diseases, multisystem proteinopathy kind of diseases. And what you see on the slide is the layout of the sequence of VCP-97 from left to right. The green portion is the N-terminal domain, the blue is D1, the purple is D2. You see that the mutations in these families are essentially located in the N-terminal domain that is very similar to what we've seen in other diseases, in multisystem proteinopathy diseases. And if you look at the biochemical readout, the signature of these mutations is just like the MSP1 mutations. They lead to increased activity in ATP hydrolysis. So that's shown in the lower right side. You see the red bars essentially show that all of these mutations, when we produce the proteins and study them, they show increased activity of ATP hydrolysis. So again, this is recapitulating what we've seen before with MSP1 mutants. And one of the other things that is again similar, but particularly striking in these Adams-Oliver syndrome VCP-97 variants, is that the N-terminal domain is so mobile that we actually cannot visualize it in the cryo-EM structures. So it's independent of whether it's bound to ADP or ATP gamma S. The N-terminal domain, the green portion where the mutations are, is so flexible and so mobile that we actually cannot capture it. So it's clearly a huge impact on what the protein does, not just in terms of activity, but this is functionally important because this green portion that's so mobile is the portion that we've heard in the previous talks actually interacts with the various cofactors that carry out the function of the cell. The obvious question is this susceptible to the well-known Cleve Biosciences drug CB5083? And the answer is yes. It is. Every one of these mutations is essentially susceptible, and the activity can be almost completely eliminated by binding to CB5083, and all of them show pretty much the same effect. We can show by cryo-EM, that's on the right-hand side of the figure, you see that we can map CB5083 to its binding site in the D2 domain. This is just like we've seen before with the wild-type P97. So no surprises here, but the take-home message from here is that indeed we can inhibit the function of the CB5083. Now, this is a somewhat busy slide, but it's pretty much everything we now know about the landscape of mutations in VCP-97, its role in MSP1, Adams-Oliver syndrome. And the left-hand side, you see essentially where the clinicians have actually captured and discovered a 7th subtype that's caused for Adams-Oliver syndrome caused by VCP variants. And these are shown to be associated with AOS. But what's surprising, of course, is that this is in these pediatric, it's a pediatric disease, and it has very similar features to what you normally see in later onset multisystem proteinopathy. On the right-hand side is now the way we think about this protein VCP-97. There's a continuous dial of changes. It's a dynamic protein. It's moving around. There's lots of bits and pieces. It's like a machine that's moving around. And these mutations, be they in D1 or the N1 or the N domain, they essentially lead to effects in this continuum of motions. But the most important message is that these are mutations that uncouple these motions. These are highly regulated molecular machines that do exactly what happens at each do exactly the right thing at each stage. But these mutations essentially introduce a level of uncertainty and flexibility that each of the bits and pieces does its own thing. So one of the things that was striking is the effect of CB5083. And I want to tell a related story, which is what happened historically with CB5083 when it was actually tested in the clinic. And this is pioneering work by Berkeley Biosciences. Their intention was to use this as a cancer drug. But strikingly, when this went into phase 1 trials, the trial was discontinued because of the off-target effect of CB5083 on PDE6, a protein in the eyes. We felt that this kind of thing happens so often in drug discovery that you have something that binds a target, then suddenly you find out later, sometimes phase 1, sometimes phase 2, that there's an off-target, and then you have to stop it. And we've seen this story play out so many times. So what we've been trying to do is to find general methods to anticipate this off-target effect. And one way to do that is to use Cryo-EM technology, which has now become much easier to do, and look at not just the structures of the on-target, but look at the structures of the molecule bound to the off-target. So in this case, PDE6. So we isolated PDE6 from bovine retina and essentially carried out the same experiment, which is add the molecule, look where does it bind. And as soon as we did the structure, we instantly knew where the problem was. We knew that if you look at the drug itself, and this is of interest to medicinal chemists in the room, there's various sections of CB5083 that were optimized to carry out its function. But if you go from left to right, there's a functionality that you see on the top left is functionality that was needed for CB5083, but actually is what was leading to the binding of PDE6. So what we did was to design, this is work done between my lab and Gandiva Therapeutics, where we designed a molecule called GND135 that actually was intended to do everything that CB5083 normally does to VCP-97, but not have this off-target effect. And this is some of the mechanics of how we did this. And over the course of this effort, we tested maybe 60 or 70 compounds and went from a very weak molecule to something that was pretty potent in about a year, less than a year. And one of the things I want to share, and you've heard a lot about Cryo-EM technology, why this really matters is as we make better and better compounds, we're looking for independent evidence that there's something happening in the binding site. And if you look at the three panels that you see from left to right, when we go from a molecule that is okay at 300 nanomolar to something that's 10 times better, you see that you begin to see much more detail in the binding site, which means that the molecule is being held more rigidly. You can see water molecule densities, and this inspires confidence and it tells the medicinal chemist that things are heading in the right direction. So this is just an example that we've seen now. We've seen this pattern pretty much in all the drug discovery efforts that we've been carrying out. And where this is right now is that if you look on the left panel, the molecule that we now have is comparable, if not slightly better, in terms of VCP-97 inhibition compared to CB5083. But the dramatic thing, of course, is we've completely eliminated the off-target to PDE6. It's gone from nearly 90% activity down to undetectable levels below 1%. So we feel that this is progressing. In vivo preclinical studies suggest that it's has very similar effects to CB5083. So again, it's just continued effort to try to anticipate things and try to get better molecules. One of the points mentioned earlier was, and discussed at length, was the context of VCP-97 in the cell binding other proteins. And obviously, if there's a way to target those interfaces, it'll be important. We have not been successful in doing that. Maybe Michelle will speak about this terrific work they're doing. But I want to share with you the general approach we've been taking to the molecular glues. And if time permits, I'd like to just share with you an example of work we've been doing in the oncology space, where we're leveraging the core idea that when there are transient protein interfaces, which is how the cell does its business, designing small molecules called molecular glues that latch onto the interface is an important way to inhibit their function. So essentially, if you have a target protein we're going after, by finding small molecules shown there in the pink in the middle that essentially inappropriately stabilize an interacting protein that normally should be coming off, but you just keep it in there, you have a way to therapeutically influence what the target protein does. So we've chosen to work in the MAP kinase pathway, which is disrupted in nearly a third of all cancers. And there are several protein-protein interactions we're looking at there. One particular one that's shown here is the interaction between a protein called RAF and MEK. They're both kinases. And the idea here is that by essentially stapling RAF to MEK, we prevent further downstream activation that happens in the nucleus. So the way we use AI is to identify exhaustively the interfaces that matter in this kind of design. There's not much time to go into this. But we are looking at finding a range of interfaces that can be targeted. We want to make sure that they can be drugged. We produce the proteins. We make sure we can show that they bind. And then we move into discovering molecules that sit at the interface. And again, here's where we use Cryo-EM. We are using Cryo-EM to ensure that the molecules we discover in our functional screens actually sit at the interface, that they have residence at that interface between the proteins. And then we move into lead optimization and to find molecules. And that has been successful with the RAF-MEK case, where we have essentially started with a really weak molecule that we think should bind and optimized it quickly to get to a potent molecule that actually compares well and does slightly better than the clinical comparator that was approved by FDA last year. Again, the value of Cryo-EM here is shown on the right-hand side. We're visualizing not just the molecule, but all the surrounding real estate of the water molecules, the side chain orientations, and that is enormously helpful in sharpening the design. We've just done recently, last year, studies in preclinical models, and what's shown on the left-hand side is in two different CDX models. The molecule is active, and it's in terms of the tumor volume shrinkage, it's certainly performed slightly similar or slightly better than the clinical comparator. So our hope is to keep moving this forward. But I mentioned this here in the context of the hope that we can actually do similar things with proteins that bind VCP-97, especially the N-terminal domain, and modulate its function. In the last couple of minutes, I want to share with you, where I think Cryo-EM technology can go. It's still a very slow technology. I've been working in this field for nearly 20 years. We feel that things need to be improved. And if you look at ideally how it can be useful for biochemists at large, there's no better place to look than just look at the adoption by the community. So 10 years ago, there were hardly any Cryo-EM structures. And in the recent review that I wrote with Werner Kuhlbrandt and Richard Henderson, we're looking at the growth of Cryo-EM as a technology compared to X-ray crystallography. And what many may not actually realize is that X-ray crystallography has kind of plateaued in terms of the number of depositions. Obviously, there's much more that's not reported, but it's a mature technology. And Cryo-EM is not quite there. It's growing nearly exponentially, now a little more linearly. And we think that the adoption will lead to discovery of many conformational states, like we heard in the previous talks. But I think the missing piece is we need automation. I think years ago, I used to dream about this. As a structural biologist, what if we didn't have to do all this business of being at the microscope? We just put some samples in, put it into a box, and then something happens, and at the end of the day, out come structures. This is in 2006. I filed the only thing the NIH let me do, because I felt this is what was needed. We worked over the years with the manufacturers, FEI, Thermo Fisher, subsequently, and JOL. And actually, in some measure, this happened. The technology is adopted widely across the world. But what this misses is we're describing structures. But what we actually want is much more than that. So we want really motion. We want to capture, oops, that's not what we want. I described the, something died here, no signal. Okay, anyway, I was just getting to the point that we really want to discover what you see on the right-hand side, maybe fleetingly, which is that we want movies. We want movies out of the microscope, and we've begun to take on this challenge of using the microscope, cryo-EM instrumentation, not just to discover structures, but to get the complete motion. So with VCP P97, ideally you want the full landscape, and we've done that with SARS-CoV-2 spike protein. And it's interesting. It's obviously needs a lot of AI, needs a lot of compute. But what we are trying to do are develop these methods for latent space analysis, where you put something in the microscope, and you get something that's instantly relatable by someone that's not a structural biologist, or even a biologist for that matter, which is you want to know what exactly is happening with this protein. So I feel hopeful that there's some advances like this may actually help us be more useful for the challenges like VCP P97. And I just want to acknowledge the fantastic team of people I work with both in my lab and at Gandiva Therapeutics. And in particular, I want to say that it's been inspiring for me to work at UBC, where my lab is in the Center for Brain Health, where many of my clinical colleagues directly work with ALS patients and people with a variety of diseases, degenerative disorders. And I think we have numerous discussions of how there may be ways to actually both measure the role of P97 and also find ways to go after it. I thank you very much for your attention.
Thank you, Sriram. We have time for questions.
Hi, great talk. So I have a question related to the Adam-Oliver syndrome that you presented. So is it a de novo or an autosomal dominant trait in those families?
This is a cohort that they identified. I think they have there's essentially 7 patients now. And I don't know, I certainly am not qualified to provide that answer, but I think they're trying hard to figure out exactly how to classify it. Because so they were looking for patients that had pulmonary hypertension, of which only a small subset have these mutations. I don't have a good answer to that, but I can certainly find out what their latest thinking is.
All right, thank you.
Sriram, I'll ask a question. I really appreciated your closing remarks about using advances in cryo-EM and AI to advance your development of therapeutics, but I'm also curious if AI can be used to also integrate observations using other biophysical tools, for example, HDX, FRET, other light microscopy images to integrate insights from orthogonal tools in addition to structure to advance therapeutics development.
Yeah, Peter, so the question really is, can AI be used to integrate multiple types of functional data all the way from structures to measurements, and not just cell biological measurements, but also patient data. And there are a number of efforts underway. I know of some that are happening in Canada, obviously much more in the US. And I think this is the fundamental limitation with AI is data. We just don't have good data. And when there is data, there's not good models that actually can extract the data to actually, and there's not enough of it. So there's not enough data. The models have not yet learned how to integrate it, but that's clearly where we need to go. And I think it will become useful when it includes patient data. I think that we need to close that gap. And there's definitely a lot of interest, not just in use in drug design, but that's only the front end. We need to know how does it work. And I think a lot of companies now, the ones in the news like Anthropic and so on, I think they're certainly thinking about things like that. So I think there's hope. And I think, as Nathan said at the beginning, we have to learn to work with AI because it's here to stay. And so the better we use it, the better we learn to use it, the better for us.
Hi. Thank you for sharing your incredible work. My question pertains to the drug you mentioned as a result of your AI, how does that relate to the, I think it's CB5339 that Cleve have designed that do not have the PD6 effect? Maybe you can speak to that, what has happened to that drug. And also I wanted to thank you for sharing this information about Adams-Oliver. As a clinician, I see patients, and it's the first time I'm hearing about its association with VCP mutations. So hopefully there'll be therapy for that disease. Yeah.
Thank you. There's a couple of questions. So let me touch on the first one, which is the relationship of GND135 to CB5339. We didn't know about this work because they had, so I think maybe perhaps they had it before. So our work was done starting from knowledge of CB5083, and there was just classical medicinal chemistry design all the way to the end. The changes we made in the end, the position of the molecule where we made the changes are exactly where 5339 also eventually made the changes. The last I heard of it, and maybe Michelle knows more about this, there was a company in China that was had taken over from Cleve Biosciences, but I have not seen anything recently about it. We don't have funding to actually push our work forward, but I'm trying hard to see if we can advance it to see, because hopefully it'll have very similar effects. But at the end of the day, I think the PD6 off target is not really a showstopper from our point of view. But we addressed it anyway. But I'd like to see it go forward. I have not heard anything about 5339 recently, but last I heard it was in China. The other question about Adams-Oliver syndrome was really interesting because we did not expect. We thought that maybe if it was seen there in this pediatric disease, it may have something completely different, but it turns out to have the same thing. So that tells me fundamentally what's happening in the cell has got to be somewhat similar. The cellular effects must be similar. And very recently, just a month ago, I had dinner with Mike Strong, who's a clinician at University of Western Ontario, and he sent me, he had seen a patient that had some variation. I think he had exactly the same stories that we heard this morning. He had not quite diagnosed. He sent me, he said that they had just done sequencing and they found the mutation P97. Within a few minutes, I told him exactly where it was. It's in the N-terminal domain. It's one turn from the residue 89. So I think this is much deeper than what we think. It's connected to many diseases. We just haven't measured it. It's just hidden below the surface. So
maybe some other modifier effect in addition to.
It's because it's so promiscuous. I suspect that it will be seen in many other diseases. Yeah.
Thank you.
Okay, thank you, Sriram. Please join me in thanking him, as well as the other speakers of our session, Andy and Po-Lin. Thank you. At this time, we'll go ahead and invite our speakers to join us here at the front, where we'll have a brief panel discussion. And we invite everyone to ask your questions, anything related to things that you heard today or learned today, and we'll take it from there.
Great, thanks. You know, one thing I wanted to ask a little bit more about was about the 701 compound. And so you said, I think whatever we think about VCP and we think of, hammers, activate it, inhibit it, maybe stabilizing it, maybe putting it in a more neutral position, or maybe, and so I guess I was wondering about the 701 compound. You were able to show that it stabilized the end UN domain, not the UN domain, but the end domains that went more down in the mutation. Does it have similar potency to CB5083, or no, sorry, to NMS in inhibition? Like, are there ways of kind of, maybe we don't want to inhibit VCP in this disease, maybe we want to just stabilize it. And I guess can you dissociate those two? So I'm not sure if I'm being very clear. I'm trying to be clear, but.
Okay, all right, thank you for the question. So I don't remember the exact number, but I know that the potency of the 701 is very close to the, I think it's even better than A73 when we have a group discussion. And then that's interesting. And also in our structural determination, we haven't found any compound. We also have other data sets. We have probably 7, 8 different compounds with the structures. But this is the only one we see 2 different entity flap up and down. And then that is on the 50-50 percentage. It's not something like 80-20. So that's made us think that maybe the drug makes something and then can modulate these allosteric pathway and then make the entity up and down and then influence its function.
But I guess my comment is the goal may not be inhibition. The goal may be stabilization that may allow interactions that are, I guess, is that a way, how do we start thinking about screening for things like that? So now we're not screening for inhibitors, we're screening a very complicated way of doing cryo-EM screens, and I mean, it sounds impossible, but.
I think it's possible now. Yeah. I think Sriram provided a very promising vision for us to keep doing the cryo imaging. I think that's a very productive way to generate the data and analyze those structures.
Tommy, before we get to you, maybe I'll follow up with a follow-up question, because I do wonder, in light of molecular glues and what Sriram just shared about stabilizing protein-protein interactions, but if you think about VCP as, itself as this, it's got multiple domains, but if you consider multiple domains as different proteins, I wonder if there's value in considering stabilizing or shifting the equilibrium with a molecular glue-like compound in a more down state, for example, for MSP-related treatment. I don't know, it's rather pie in the sky, but just a thought.
I have a microphone. Yeah, no, I think the way you framed it, Chris, I think that's exactly the question. Inhibition is a hammer that we don't want to hit it with. It seems to be the only thing we know how to measure. And the question is, how do we navigate our path from knowing how to inhibit it to, I wouldn't even say maybe stabilize it, I would say to control the types of interactions that we want to be favored and dial out some of the other ones, which is where I think the molecular glue approach might be helpful. So if we go after the inhibition, which happens below on the D2 domain that Po-Lin spoke about, and then the action is somewhere else, I think we just have not yet found the right kind of tool. But I think the way you framed it is probably the way we should think about it, which is how do we find ways to modulate selective activities and keep them on, as opposed to shutting the whole thing down, which may not be a good thing to do, despite the fact that ATP hydrolysis is really elevated in all these mutations.
Maybe I can add to this real quick, how important it is to consider the time that individual cofactors spend there and how this is distributed. And at the moment, we don't have enough insight yet, because the systems we study in detail are not complex enough. We usually don't have competition between different cofactors to see what the distribution is. And just as an example, a few years back, we had characterized a few MSP mutants together with Ray Duchesne, and they showed changes in the orientation of the N-terminal domain, so it was more up, and the UN cofactor, for instance, bound 10 times more quickly, so the on rate was strongly affected, but we don't have the information how this has an influence on other cofactors, how this is now to the disadvantage of other cofactors who would like to use the same pool of P97, so that's why fine-tuning this and then knowing what this really means with up and down and under the active conditions in a complex soup, the soup of the cell with all these competing processes. They all want to tap into P97 as a motor, and so I think that's the challenge at the moment. Still lack of information about that.
Yeah, so I was really fascinated by your work in Adams-Oliver syndrome, and especially given the exact same residues affected in MSP are now causing a lung and skin disease as well. So two questions: is it known whether there are actually ubiquitinated protein inclusions in the lung and skin in that syndrome? And then secondly, any idea based on the residues and and exact changes as to any specific, interactors or functions of VCP that are, altered in that syndrome.
Yeah, I wish I could answer. That question. So we just don't know. The paper on the Adams-Oliver syndrome is in press. Hopefully, it'll be published. This is just the first cut. Very little cell biology has been done. We've looked at the presumption was that it would have an effect on notch signaling, but none of the mutations showed any difference in notch activity, so that remains a question mark. The only tangible handle we have is this increased ATPase activity and the location of the residues with the mutations. Again, it's so heterogeneous. Out of the mutations, there's only one that showed actually a measurable difference in stability of the protein. So there's a mutation to, I think it's arginine to tryptophan, I forget exactly what it was. That's more unstable. But the others, they're different. I mean, they're similar to wild type. So we just don't, it's doing something. And all the instincts I have is that we are not focusing on where the action is. We're looking where we know how to look, but not where the action is, which is probably at the interface between the N-terminal domain and some specific set of effectors.
I think it's really important because those mutations are similar to MSP mutations. I mean,
yeah.
They cause MSP as well. So.
Really similar, same location of the protein. So it's plausible that there are shared cellular proteins effectors that bind there. There's one of 60 or 70, we don't know how many, that actually talk to P97. So this idea of mapping those interfaces and knowing which ones to tune in, which ones to keep, which ones not to keep, I just don't know how to do that. I think this is a challenge.
Coming back to this idea of a molecular glue, and Sriram, I think you were just getting at my question. How do we figure out how many different interactions we might need to target? Is it going to be enough to maybe target one interaction, or are there going to be a handful that we want to figure out how to target potentially in VCP MSP?
Maybe Michelle will touch on this in the future, but my view is we don't have the time to do this exhaustively. We just need to shotgun it. I think we just need to just do it, get the answer, and then we'll work out backwards what it did. So I think my view would be, and that's where the AI piece comes in, that we just don't think too hard about it, just throw everything we can and then look for the effects. Because I think it's so complicated that if we did this really systematically, it may just take too long. So I don't know, maybe it's just, I think a lot of people need to work at this, but I feel that speed is everything here. So I think it seems too complicated to do this the way we might do a traditional biochemical campaign.
I think this is something that AI has to help with. If you think about the progression of the VCP structural biology over the past 10, 15 years, 2016, Sriram published the structure of P97 in Science, 2.2 angstroms, but that was just VCP on its own. And then in subsequent years, probably took another 5 years or so to get a structure of VCP, high-resolution structure of VCP bound to one cofactor. And today we heard a beautiful story of VCP bound to FAF1, and then in addition to knowing that it can also bind simultaneously to P47, the complexity is really astounding. And to Sriram's point, I think it would take way too much time to characterize individual adapter VCP interactions, especially since a single hexamer has 6 available binding sites. And so this is something that I'm excited about the possibility of AI and really helping with.
Is there time for a question? Yeah. I have a question for Andreas and maybe Peter, and it's going to sound a bit crazy. So when I look at all of your studies, they're so beautiful, right? Like you can reconstitute these things. You have 5 ubiquitins that unfold. It's beautiful. And then you look in cells, and it's a complete mess. So from your perspective, what is that transition from your studies to, the cell biology studies? And if you had advice for cell biologists, you're like, do this damn experiment. What would that experiment be?
The first thing that jumps out to me is I view cryo-electron tomography as a beautiful marriage between structural biology and cell biology. And for those who aren't familiar with the methodology, it's using the same electron microscope that we used for imaging individual protein particles, but using that to image regions within cells. I don't think the technology is quite there yet to look at the complexity of P97 complexes in cells, but I think there's a future where that's certainly the direction.
Yeah, certainly tomography, but also even fluorescence-based studies, because P97 interacts with so many cofactors in different areas of the cell. I mean, it's something that we also many times don't consider, is that this is not just one big bag where everything is the same throughout, but we have different local environments where P97 may interact differently, like near the endoplasmic reticulum. Its cofactor pool is certainly different than if it's somewhere else, and in the nucleus having a bunch of roles there with DNA repair and all that. So having information about what VCP works with, what cofactor pools it work with in different areas, and also going back to finding potential cures, I think MSP related disease is so complex that there may be a bunch of different processes that are affected by different mutations. So presumably there will not be just one compound or a couple or a handful. Maybe there need to be different ones because there are a bunch of different pathways that are affected differently by these different mutations depending on how this affects. I don't think it's a single cofactor that we have to identify that is primarily affected by the entomonal domain moving differently or being more up or down, but there may be different flavors. And going back to earlier questions about residence time, cofactor coming and going, and related also to the ATPase activity. I mean, we have compounds that may affect the entomonal domain position, but at the same time, you don't want to have inhibitors of the ATPase activity, but many times this is at the moment still coupled. So yeah, maybe molecular glues. I know a couple of years ago, Ray Deshays represented the entomonal domain movements with his arms up or down, and he said maybe we just need something that sticks in the armpit and keeps the arm down without affecting other things that the motor does. So I think that's the challenge in all this.
You mentioned that P97 and UN complex is less active for unfolding the substrate compared to the yeast variant of CD48 UN complex. Why do you think it is so? Like, there is any specific reason? You mentioned about the ATP thing. What is that about, like higher ATP level and something?
Yeah, it's really baffling how much more also the yeast homolog is turning over ATP. It's in the thousands. It has a rate. It gets twofold stimulated, so it idles with 1,000 per minute, and then when its substrate is engaged, it goes to well over 2,000 ATPs per minute. And the P97 VCP is much slower on that. So it could be that it is more efficient in catching a substrate because it takes more attempts. It may move more quickly through different states, and so even if it fails a few times of kind of capturing a ubiquitin that is presented by the UT3 domain or by NPO4, but it takes more attempts and more rapidly to really grab it and then suck it through. So yeah, it may have to do with the dynamics of it. I think the cofactor orientation on top of the motor and how well defined it is, I think that's equally tricky for both. It's equally dynamic because even for the CDC48, people have been struggling visualizing, especially the UT3 domain of UFD1, because it's rather mobile on top. So that is similar between the human and the yeast system. But the dynamics of the motor overall, how quickly it fires, how quickly it moves, is quite orders of magnitude between that. So that may explain the difference. But we also see, for instance, just to extend this real quick, we see that also for the proteasome. Proteasome in yeast is much more kind of less selective in how it binds its substrates, and it switches conformations much more rapidly. It has a higher idling rate of ATP consumption, whereas the human proteasome is very strictly regulated. It has a lower ATPase activity in the absence of substrate. So it may be similar on P97, that the evolution set it up more thoughtfully and it's more tightly controlled, but there may be these cofactors that you need, and we haven't discovered some of them, that boost this activity, that really get P97 VCP to where it really needs to be, but that also makes it vulnerable to these mutations because they mess up this fine-tuned system of cofactors coming and going. We shouldn't envision this as a static system, and it was, Tarun had asked a question about this. We think that certain cofactors may deliver a substrate, and then it switches to other cofactors that are bound to process the substrate, and then maybe the release is yet facilitated by another cofactor, so there's a coming and going constantly, and it's quite complex.
I'll just say that we are what's keeping you between lunch, so we'll have time for, I think I see one more question. So we'll do one last question.
Okay, so I was sort of musing on what was said about using AI, and it seems to me that given that there's a complexity of interactions, and in humans, clearly the mutations manifest in one mutant manifests in different ways, suggesting that there's a sort of polygenic context, right? It might be that there's polymorphs in other factors, but it made me think about, is it possible to start to use tool compounds coupled with something like mass spec to generate? I was thinking about, how do you generate the data sets for the AI? And is that something that you're thinking about? In other words, that you nudge the molecule in one direction or another and then look at the constellation of factors that are adjusted, and to try to develop sort of a ground truth about how individual mutations are working, and thereby to reverse that, with drugs. I don't know if that's clear. Is that something that's already people are doing, or
No, I'm not aware of studies, but it would be nice to do extensive mass spec studies with these mutants, for instance, and see how the occupancy of the VCP is changing as you have different.
And the nice thing is there are tools now that allow you to do that more dynamically.
Really large-scale mass spec, for instance, in pull-downs or cross-linking. But I'm not aware of someone doing that, really.
No. Yeah, the mass spec data, I mean, there are other examples where mass spec has been used exactly the way that you described. But here, I feel that the use of AI that might be most relevant is to basically use AlphaFold and all of its derivatives to look exhaustively at every possible interaction, and then do some of this in silico to at least begin to whittle down the landscape. So I think something like that would have seemed like a science fiction or almost a nonsensical thing to think about, but I think that may then define what are the best experiments to be done. If someone asked about what should a cell biologist do, for example, right? So we can generate the data with mass spec, but maybe there's a cellular assay that can be done once we know the identity. So I think at least thinking about this, I feel we need to whittle it down to at least finding, okay, what are the subsets? What is a subset of proteins that we should worry about? And then think of it more holistically, both maybe mass spec, maybe HTMS, and maybe cell biological readouts. I think it hasn't happened. So I think maybe this community could actually come together to lead that effort to at least define the problem more clearly. So right now, I think that's still a little bit vague.
Well, it would seem in the first instance, you've got 100+ mutations, right? Each one of them is going to create a distinct constellation of interactions. I mean, that you don't even, that's a starting place, and then you don't have to choose 100 different conditions. You can pick the ones that are related by domain and so on. So narrow it down. I would think that it was tractable.
For any particular, you're right, it's tractable once you pick the modality, for example, mass spec or whatever. Yeah, so I think that's, I think anything that produces credible data is going to be helpful. So that's certainly one approach.
Okay, with that, we'll go ahead and conclude our session. Please join me once more in thanking our speakers.